If calculations, diagrams, and cancer biology make you nervous in exams, stay with me. In the next few minutes, I'll show you how question 8 of this GCSE AQA Biology Paper 1 for higher triple science students from June 2024 can actually become a scoring question if you follow the examiner's method. Hello everyone, and welcome back to my channel, DB Tuitions. This is your tutor doctor. Now in this video, we are covering question 8 from the AQA GCSE Biology Paper 1. I want to be very clear before we start. Every answer you will hear in this video is taken directly from the official AQA mark scheme. That means you're not just learning biology, you're learning how to score marks. And let's go step by step. Let's look at the question 8.1. This is a nested question. As in previous videos I've explained, a nested question is a question in which, let's say this is question 8, but you will have 8.1, 8.2, and so on. So if any data given in any question can be applicable and be useful in answering whole of that question. So let's read the question, and 8.1, always eyeball how many marks is it for before you read the question. So you set the expectations that this is a 1 marker, 1 sentence, couple of words, or even maybe 1 word, depending on what the question is. Now, getting into the question, cancer is caused by changes in cells that result in uncontrolled cell division. Before a cell begins to divide, its DNA replicates to form 2 copies of each chromosome. Describe one other change that occurs in a cell before the cell begins to divide. Now, please do remember, it's specifically asking you for one change before the cell begins to divide. Now, before a cell begins to divide, it must prepare itself. And that one correct change is that the cell increases in size, and also the number of subcellular structures. So these instead of just writing subcellular structures, you can pick up anything. You can say ribosomes, mitochondria, or any subcellular structure. Now remember, only one change is needed. Writing more does not give extra marks. And because of this, when the cell splits, both the daughter cells are going to get equal amount of subcellular structures. Very well done. Now let's move on to question 8.2. Again, that is a 1 marker question. And can you see how there's a figure 8 given for reference? 8.2 question reads, name structure Z in figure 8. And if we can look at figure 8, Z is the cell membrane. It is pointing out to the cell membrane. Now, it's very important that whatever label line you're looking for, in this case it is pointing to Z, the outer boundary of the cell. So therefore we said this is an outer boundary of an animal cell, that is a cell membrane. Now always follow the label line. If the arrow touches the edge, the answer is membrane. And let's say the arrow is going in and pointing towards that, then the answer would be chromosome. And that was just for the example, but in this question it's clearly pointing to the cell membrane. Now let's move on to question 8.3. Now again, this is a 6 marker. There you go, we came to a 6 marker question. 6 marker questions can be calculation questions, or they can be command words you need to remember, explain, describe, evaluate. So again, please follow this question step by step. If you want, you can pause the video here, try to solve it by yourself, and later come back and check. Now let's read the question. Structure Y in figure 8 is a cylinder. Now, for structure Y, the real volume is given in nanometer cube and the real radius is given in nanometers. And the length of a cylinder is calculated using the equation length equals to volume over pi times radius squared. The length of the image of structure Y in figure 8 is 4 mm. Calculate the magnification of structure phi in figure 8 using the pi value is 3.14. Now, formula and the given values, we need to note them down. Now, this looks like a long calculation, but this is actually very structured. Now, first, let's calculate the real length using the formula given. Now, first, let's simplify the denominator in this. I'm going to do pi times radius square. So 3.14 times, I'm going to square the 125. So I'm going to put that into the calculator and my answer is 49062.5. Now let's take the volume given and divide it by the pi r square we got. So therefore the length, substituting the values here, 2, 4, 5, 0, 0, and then 3 zeros. Be very careful with the amount of zeros you're putting in. Make sure you count them properly. And therefore, that's approximately, when I put that in my calculator, it is giving me 499 nanometers. Now, remember, the length of the image of structure Y given in figure 8 is 4 mm, but I have got the length in nanometers, so I have to convert my nanometers into millimeters. So that's very important. Now, converting my 499 nanometers into millimeters, I'm going to get 0.000499 millimeters. Now, the next stage is magnification. Now, remember, magnification equals image size divided by real size. So, magnification is equal to the image size divided by the real size. Now let's go ahead and calculate that. 4 over 0.0000499, and when I do that, I approximately get around 8,000. So the magnification is approximately 8,000 times. Now, even if your final number is wrong, showing the method still will earn you marks. So please make sure that you write down, once you've calculated, please do write down the answer in the given space. And remember, magnification has no units. If you pause the video and you got the right answer, absolutely very well done. Now let's continue to the next question. And again here, there are some figures given, which is figure 9. We can see stage 1, stage 2, and stage 3. These are the stages of cell division. In stage 1, we can see that there's fibers attaching to the replicated chromosomes. Then we move on to stage 2, and in stage 3, the fibers attached to chromosomes and they're getting shorter, pulling the chromatids apart. Now let's read the question. Some cancer drugs prevent cell division. Drug X prevents the fibers from attaching to the replicated chromosomes in stage 1. So remember, if the fibers cannot attach to the replicated chromosomes, they cannot pull the chromosomes apart to opposite ends of the cell. It means the cell division cannot progress. So therefore, when they say explain why a cell cannot complete division when affected by drug X? This is your question 8.4. Now, what we are seeing, these fibers are known as spindle fibers. Now, drug X is preventing the spindle fibers from attaching to the chromosomes. So this is what it is already telling us. That's what the drug X is doing. So because of that, we mentioned that because of this, chromosomes cannot be pulled apart. We are going to write that cannot be pulled apart. So the cell cannot complete the cell division. Therefore, or we can also write it cannot complete mitosis. Either one is fine here. Remember, key phase here for this question's answer is chromosomes are unable to separate because of the effect of drug X. Very well done. If you've gotten so far and gotten everything right, you're right on track. Let's move on to question 8.5. Give the reason why a drug that stops cell division helps to treat cancer. Now again, this is a one-marker question. Cancer cells divide rapidly. That's how cancer grows. So cancer is caused by uncontrolled cell division. Drugs that stop cell division slow down. So the key phrase is they slow down or stop the tumor growth. That's very important to mention, which helps to treat cancer. So therefore, the drug is stopping the cell division, slowing down or stopping the tumor growth, which is helping in treatment of cancer. Now moving on, the next question, 8.6, again is an MCQ for one marker. New cancer drugs are tested in clinical trials. Preclinical testing happens before clinical trials. What is involved in preclinical testing of drugs? Now, preclinical testing happens before drugs are tested on humans. At this stage, drugs are tested on live tissues in a laboratory to see how they react. So therefore, this is our correct answer. Now remember, side effects and optimum dosages are tested later during clinical trial once we have established that that drug can be used on a live tissue. So that's why it is very important to look at all the options and eliminate the ones which don't seem like. That gets us to the end of the question 8 and also the series of the entire paper 1, in which I have done 8 different videos for each question. Now let's quickly recap this question 8. So the key content we have learned in this is cells prepare before dividing, not only the DNA, but also the subcellular structures. They grow in number so that they can be distributed into the daughter cells. Now, always when labeling, follow the arrow to the edge. In this case, it was pointing towards the cell membrane in question 8. Now, magnification equals to image size divided by the real size. And always make sure whenever in calculation, your units must match. And cancer is nothing but uncontrolled mitosis. And remember, preclinical testing uses live tissues. Now, as we have completed question 8, and with that the entire AQA GCSE Biology Paper 1, June 2024 fully explained. Every answer in this video is mark scheme approved. You can confidently use this as a model for exam writing. If this video has helped you, please My sincere request, subscribe to our channel DB Tuition 09 so we can make more content and freely share with all of the students. Please do share it with your friends if you like the explanation, and check out the rest of our question by question exam series. As soon I'm going to get out the paper 2 and again break down it into question by question. Remember, exam success is not about writing more. It is about writing what examiners want, and I'll see you in the next video with paper 2 content. Take care.